GCSE Biology for WJEC and EDUCAS. Cells and enzymes is the very, very beginning topic. So, cells. Today's cell theory states that all living organisms are composed of cells. I'm turning up the light because it's hard to see. Oh, they're turning down the light. Ooh, yeah, that's better. All living organisms are composed of cells. They may be unicellular, which means one celled, or multicellular, many cells. The cell is a basic unit of life. Cells are preformed from pre-existing cells during cell division. Energy flow occurs within cells, enabling the chemical reactions that make up life to take place. Hereditary information, which DNA, is passed from cell to cell when cell division occurs. And then other ones, all cells have the same basic chemical composition, although all cells have features in common. There are also differences between different types of cells. Some of those differences allow scientists to classify cells as either animal cells or plant cells. So, plant and animal cells, they all have the cytoplasm, which is the site of most chemical reactions. Cell membrane controls the entry and exit of substances, what enters and leaves the cell. Nucleus contains chromosomes which carry genetic information and controls the activities in the cell, and mitochondria is the site of aerobic respiration, which supplies the cell with energy. In plant cells, they have features that animal cells don't have. Cell wall made of cellulose, which is a structural support. The sap vacuole, which contains a watery sugar solution, which is sap. A swollen vacuole pushes the rest of the cell contents against the cell wall, making the cell firm. In plant cells, cell vacuoles are large, permanent, and central spaces. And chloroplasts, which are the site of photosynthesis, they absorb light plants, and chloroplasts absorb the light plants need to make their food by photosynthesis. They're not found in all plant cells, but they are never found in animal cells. So it's only plant cells you see them in. So you need to be able to remember the nucleus, which looks very similar in both diagrams, the mitochondrion, which is just a wiggly thing, cytoplasm, cell membrane. And then you need to be able to label the chloroplasts, the mitochondria, which is plural, the sap vacuole, the cell membrane, and the cell wall. So, summary, animal cells have nucleus, cell membrane, cytoplasm, mitochondria. Plant cells have all of those, and as well chloroplasts, cell walls, and vacuoles, although only some have chloroplasts. It also depends what type of the plant, what area of the plant you're looking at, because roots don't need chloroplasts, they're not necessary. You can observe cells through a microscope, and you just need to know different parts of the microscope. So the eyepiece lens has a fixed magnification, although it is possible to exchange it with a lens of a different magnification. So that is the part at the top. The objective lens has different magnifying powers that are interchangeable. They're the ones that you use to adjust the magnification of the image that you see down the microscope. So these are the objective lenses, they're the ones you just push around and flick through. At the stage where the microscope slide you are observing is placed on, and it has some clips to hold it in place, the iris diaphragm can be opened or closed to adjust the amount of light entering the objective lens. You have the condenser, which is sometimes present. It concentrated the light into a beam directed precisely into the objective lens. You have some older microscopes may have a mirror, which is used in conjunction with a separate lamp to shine light through the condenser and iris diaphragm. Have a coarse focus to just get it to roughly in focus using the lowest power objective possible, which is here, it's a bigger one. And then you have a fine focus used to fine-tune the image and make it as clear as possible, which is by here. And you have the microscope slides, which is just they hold thin specimens or sections. Biological stains can be used to allow more detailed cell to be seen. To calculate the total magnification, you need to multiply the power of the eyepiece lens, the top bit, by the power of the objective lens. So 10 times 4, 40. 10 times 10, 100. 10 times 40, 400. For microscope drawings, you need to use a sharp pencil. You need thin, clear lines and that don't overlap where they join. Do not shade parts of your drawing unless it's absolutely essential in order to clearly distinguish structures. Always use a ruler for labeling lines, and the lines should never cross each other. Don't label on the drawing itself. Keep the labels outside the drawing, so if you draw something here, just put a line over there and then label it. You need to make sure the drawing proportions are correct, and so if the structure is twice as wide as it is long, then the drawing should be like that too. And also, it hasn't mentioned it here, but usually they want you to do the labels in all capitals. There are some specified practicals, so the examination of animal and plant cells using a microscope and production of scientific labeled diagrams. There's also looking at cheek teeth cells, where you just scrape the inside of your cheek with cotton wool bud, smear the saliva from the bud gently onto your microscope slide, add a drop or two of water to the part of the slide you've smeared, and then place the cover slip onto the slide. You place one edge of the slide, you place one edge on the slide and then gently lower the cover slip down, trying to remove the bubbles. Add some methylene blue dye near one edge of the cover slip on the microscope slide. You draw the dye under the cover slip by putting the filter paper next to the opposite edge of the cover slip with the dye. Leave for a few minutes for the dye to stain the cells, and then you look at it under a microscope. Yeah, and you just stain it so you can see the cell more clearly. There's also an onion cell one, where it's very similar. So you split and pull the onion part into layers. Make a square cut partway through an onion segment, and make sure it's on the side of the segment that was towards the inside of the onion, and that the square cut is smaller than the cover slip. You use 4 steps to peel the inner epidermal layer away from the onion, so it's just like a really, really thin skin, which is on the inside and outside of each of the layers of the onion. And then you place a drop of iodine stain onto the center of the sheet. Gently lay the sheet of epidermis onto the drop of dye. Try to avoid trapping air bubbles underneath the tissue. Lay a cover slip over the tissue, leave it a few minutes, and then you can look at it under a microscope and make a drawing. There are some specialized cells. They have specific jobs to do. So, for example, you can have sperm cells, which has very small cytoplasm, it has a tail that swim fast. Red blood cells, they've lost their nuclei, so they can be more packed with the red pigment hemoglobin, which carries oxygen around the body. There's also xylem cells, which form tubes which carry water up the plant and also strengthen the plant. To do this, the cells have perforated end walls. The cell wall is very thick, and the cytoplasm has died off to leave a hollow tube. Within the body, the organization of things is different levels. So there's a tissue, which is a group of similar cells with similar function, like blood or xylem or bone or muscle. And then an organ is a collection of several tissues that perform a specific function, like kidneys, brains, hearts, leaves, flowers. Organ systems is a collection of several organs that work together, like the digestive system, the nervous system, respiratory system. And you have organisms, which is a collection of organ systems to make a whole animal or plant. So, like cat, people, rosebush, oak tree, coconut tree, whatever other tree, fish. A bone is an example of a tissue. It's made of 2 types of similar cells which together form and maintain bones. A bone is an organ as it contains both bone tissue and blood. Leaf is another example of an organ. Several different types of cell which performs different functions, all of which is linked to the production of food, photosynthesis. So this would be a vein, which contains xylem tissue to bring water to the leaf, and phloem tissue to transport sugars away from the rest of the plant. The upper epidermis, which transparent skin, which lets light through. Palisade layer, lots of chloroplasts to absorb light for photosynthesis. Spongy mesophyll also has chloroplasts for absorbing light. Lower epidermis forms the outer skin of the leaf. Guard cells changes the shape to open the gap, which is called the stoma, to let carbon dioxide in for photosynthesis. Enzymes. All of the activities of a cell depend on chemical reactions. It's been estimated that 10 million reactions occur in a typical cell every second. These reactions are controlled by special molecules called enzymes, which enzymes are produced in cells is controlled by another molecule, DNA, which is found in the cell nucleus. So, enzymes are proteins made by living cells which speed up, catalyze the rate of chemical reactions. So, enzymes are involved in all metabolic reactions, building large molecules from small ones, as well as breaking down large molecules into small ones. Enzymes act as catalysts, which, as I said, speed up chemical reactions. They remain unchanged by the reaction it catalyzes, and they're specific, which means that only a certain enzyme will catalyze one reaction, or only catalyze one reaction or one type of reaction. The chemicals on which enzymes work are called substrates. The part of the enzyme which binds to the substrate is called the active site. It is held in shape by chemical bonds. In order to catalyze a reaction, the enzyme has to lock together with its substrate, so they have to be complementary shapes. The shapes of the enzyme's active site and substrate must match, so that they fit together like a lock and key, or they're described by being complementary to each other, like puzzle pieces, for example. In the same way, the key fits into a lock, so that the substrate can fit into the active site of the enzyme. That is why enzymes are described as being specific. They can only work with substrates that fit into their active sites. In some reactions, an enzyme catalyzes the breakdown of a substrate into two or more products, while in others, enzymes causes two or more substrate molecules to bind into one product molecule. So, breakdown of protein into amino acids, building of amino acids into proteins is another one. And when a enzyme binds to a substrate, that's called an enzyme-substrate complex. Enzymes work better as temperature increases, but if the temperature gets too high, then they are denatured. Different enzymes are denatured at different temperatures. Enzymes work best at particular optimum pH values, which is different for different enzymes. So increasing the temperature of a reaction makes it work faster to begin with, because the enzyme and the substrate molecules move around more quickly as they have more kinetic energy. This results in an increase in successful collisions between enzyme and substrate molecules, forming more enzyme-substrate complexes. Enzyme action will be greatest, and therefore the reaction rate will be highest at the enzyme's optimum temperature. But once you go past the optimum temperature, the enzymes stop working altogether. The high temperatures break the bonds that hold the enzyme's active site in its specific shape, so too high of a temperature would break those bonds and destroy the shape of the active site, so it can't form enzyme-substrate complexes anymore, because it can't bind to the substrate can't bind to the active site, so the enzyme is said to be denatured. This is an example of just over temperature, rate of enzyme activity to a point. That is the ideal temperature, but then past optimum temperature, the enzyme are denatured. The actual temperature that denatures enzymes is different for different enzymes. Some enzymes start to denature about 40 degrees Celsius. Most denature at around 60 degrees. Boiling is fairly certain to denature an enzyme. A very small number of enzymes, mostly found in bacteria, have been found to tolerate temperatures as high as 110 degrees Celsius. Most enzymes can't, though. If you want to test if a certain sample is acting as an enzyme, they do a control experiment in which a boiled sample of the substance is used in place of the unboiled sample. The reaction doesn't occur in the control, then it's assumed the substance acts as an enzyme because the boiling process has denatured it. pH has a similar thing. Unsuitable pH conditions can break bonds that hold active sites in specific shapes. This changes the shape of the active site so that the substrate molecule will no longer fit. Enzymes work best at their optimum pH, but if pH is increased or decreased away from the optimum, the active site changes shape, meaning the substrate cannot bind and the enzyme is denatured. So, if you want to denature an enzyme for use in control experiments, the most effective way is to boil it. Altering pH away from the enzyme's optimum pH will also denature it, though. High tier thing, enzymes as proteins. Proteins are long chains of amino acids linked together. Different proteins are composed of different orders of amino acids within the chain. The long chain of amino acids is then folded to form specific shapes, e.g. the active sites of enzymes. If the amino acid sequence changes in the chain, the protein will fold into a different shape structure and won't be able to form its function. The effect of boiling on the activity of catalase in the liver, it is just another investigation. You look at an enzyme called catalase. Catalase is found in many tissues where it breaks down harmful hydrogen peroxide that is produced by reactions in cells. You need your risk assessment, so scalpels are sharp, so you may cuts when using scalpel to cut the liver may occur, so you cut away from the body onto the white tile. Boiling water can scald scalds from hot water when boiling water. So you need to run the skin under cold water immediately if you scald yourself. Hydrogen peroxide is harmful. It could enter your skin and be transferred to your eyes. Get onto your skin, sorry. So you just need to wear goggles. So you cut 2 pieces of liver so that they are about 1 centimeter square, to make it a fair test, just to keep everything controlled. That's one of the controls. You place 1 piece into a test tube containing boiling water for 2 minutes to kill all the cells, and you pour 20 centimeters cubed of hydrogen peroxide into each of the 2 test tubes. Place the uncooked liver into 1 test tube and the boiled liver into the other. So the tube containing the unboiled liver produced lots of bubbles, so the gas produced was relit with a glowing splint, therefore we know it's oxygen. The tube containing the boiled produced no bubbles, so the splint did not relight. That's because the unboiled liver contained catalase enzyme, which catalyzed breakdown of hydrogen peroxide into water and oxygen, so that's why oxygen was present. In the boiled piece of liver, the catalase enzyme had been denatured by the boiling process, and so the reaction didn't occur. No oxygen was made. There's another one on the pH with potatoes. You just sort of do a very similar thing. You have 20 potato discs added. Let me read through it. In the previous experiment, you used liver as your source of catalase enzyme, but catalase is found in many other types of tissue, including plant tissues such as potato. It has the same function in all tissues of breaking down hydrogen peroxide into water and oxygen. One of the products of the reaction is oxygen. The activity of catalase can be measured by collecting oxygen produced over a set time, like 5 minutes. An investigation was carried out using potato as the source of catalase. 10 centimeters cubed of hydrogen peroxide was placed in the reaction tube, together with 5 centimeters cubed of a pH 3 buffer. 20 potato discs were then added, and the volume of oxygen collected in 5 minutes was recorded. The experiment was repeated at the pH values 3, 5, 7, 9, 11, and 3 times at each pH value. So, as the pH value increased from 3 to 7, the volume of oxygen collected in 5 minutes increased. but as the pH increases from 7 to 11, the oxygen value, the oxygen collected in 5 minutes decreases, which shows the effect of pH on the activity of an enzyme. Best temperature to wash your clothes depends on whether you're using a biological washing detergent. Harder stains to remove can be caused by lipids, such as oils and butter, or proteins, including enzymes that break down lipids or proteins in biological washing detergents can help remove those stains. Egg yolk is a good stain to test this with because it contains mostly protein with a small amount of lipid. Biological washing powders have to be used at lower temperatures because if the temperature is too high, the enzymes become denatured. Biological washing powders are therefore more economical because less electricity is used to heat the water. That is the whole topic. That is all of cells and enzymes.